Hello everyone, and welcome to the first lecture of this semester. Today I'm going to be introducing you to the scientific method, a process which you're going to be applying throughout the course of this class in your lab. I'd like you to be able to formulate a hypothesis and evaluate the quality of that hypothesis through the use of vocabulary as well as an understanding of the scientific method. You'll independently identify both the independent and dependent variables, control and experimental groups, as well as to list those experimental controls and variables. This is going to be done for each experiment, and I can give you any type of experiment, as well as to recognize that hypotheses can never be proven true or proven false, and understand that this is based on the fact that we can never account for every variable in a given situation. I'll be starting each lecture this semester with a slide similar to this, where I outline for you my expectations for the material that's included. The lecture that follows will help to give you a foundation and some guidance on that material, as well as exactly how you'll be tested. But you should be using your textbook as well as other additional materials to help you really flesh out that understanding and to try and put it into a larger context. I want you to know that vocabulary is a big component of this course. It helps to ensure that scientists know who is talking about what. And that use of vocabulary will be not only a critical component in my grading, but also a material that you need to have in order to better understand this course. So understanding root words is one way that you can better help yourself to grasp vocabulary. I will never test or grade you on these root words, but you being able to apply them will help you to better understand them. So for example, if you know that saccharide is a word that means sugar, you only need to memorize that particular root word. Then you can apply some things you already know. For example, mono means one, di means two, and poly means many. So a monosaccharide is a one sugar molecule. A disaccharide is a two sugar molecule, and a polysaccharide is a chain of many sugars. There's a list online that I've posted for you. It's a reference material list. It's not something you need to memorize. As a matter of fact, I don't want you to memorize them. But I am going to ask you to apply them in this week's first recitation, and you'll continue to see these not only in recitation and in lecture, but also in lab. So here's some examples of how you could apply this. Monomer. Well, mono means one or single, and mer means part. So a monomer is one part. And if we were to take a monomer and put two monomers together, you get a dimer. Or if you put many together, you'd get a polymer, because poly means many. Hydrophobic. Hydro meaning water, phobic meaning against or anti. This is a molecule that does not interact with water. And cytoplasm, where cyto means cell and plasm means formed into. It's the material that actually forms the shape of the cell. So these are going to be some vocabulary words we'll see over the course of the semester, and this is an example of how you can break them down. I highly recommend that you print out the PDF that's provided to you, and when you struggle with a vocabulary word, try to better understand where that word comes from. Let's take a look now at the process of scientific discovery, more commonly known as the scientific method. The core of the scientific method is the ability of scientists to develop a hypothesis, plural, meaning hypotheses. So these hypotheses are going to be critical for a scientist to develop predictions that they can test. That means that these have to be testable. This means that it's capable of being supported. For the fact that a hypothesis is testable doesn't mean it will be supported, simply that it can be. If I develop a hypothesis that mice ride dinosaurs, this isn't testable because there simply are no dinosaurs for me to run this test on. But it also needs to be falsifiable, which says that it's capable of being unsupported. If my hypothesis is that God is the all-being creator, well, unfortunately, I can never disprove that hypothesis. It's not capable of being unsupported. And so we have to have both. In order for a scientific hypothesis to be a reasonable component of exploration, By the way, these are not questions, these are predictions. It has to be something that can be both tested and supported, as well as falsifiable and unsupported. Once we've developed this hypothesis, we need to determine what controls are necessary. And this starts by better understanding variables. Variables are going to be things changed between groups, things that we can't keep the same. The things that we can keep the same are known as controls. Now, it's going to be very important to define those controls, because when we control for something, meaning when we keep things the same between the groups in our experiment, we remove those as factors that influence what's happening. The problem is that we can never account for every single variable, meaning we can never control for every single variable. So we also need to better define the variables that we're testing. So let's take a look at this. A controlled experiment compares an experimental group with control groups. So experimental controls are factors that are kept consistent between these groups and thereby remove them as variables. Factors that change as the result of the experiment are variables, and there's two that are critical. Now, remember, we can never control for every variable, and there may be things that change intentionally and unintentionally. But a good experiment starts with identifying what we're changing and what we're measuring. The thing that we're changing is called an independent variable. The thing that we're measuring is the dependent variable. I like to remember this because if something is dependent on something else, that means that it's changing because of something else. Well, the dependent variable changes because of the factor that the researchers changed. So the thing that's independent would be the thing that the researcher changes. Now, a high quality hypothesis is going to clearly define all of these. It's testable, which means that there's going to be an independent variable that changes, and it's falsifiable, meaning that there's a dependent variable that we can measure and see that change. And if that change doesn't appear the way that we predicted, that would be something that was unsupported. So in order for us to really develop a strong hypothesis, it should clearly outline all of these. What are we controlling for? What are the variables, both the independent and the dependent? So I understand that this is a little bit twisted. It's hard to get your head entirely around. I'd like to start by just providing you some examples. The administration of an anti-cancer drug will reduce the volume of the tumors more than the placebo. That is my hypothesis. Now, my experimental group is the group that gets the new anti-cancer drug. The control group is the one that gets the placebo. And you'll notice that this is clearly outlined in my hypothesis. I've also clearly outlined that the independent variable is the administration of the drug or not. Note that that's what's different between my control group and my experimental group. The dependent variable would thus be the impact the drug has on cancer. In this case, the volume of the tumor, which has also been described. Let's take a look at another one. Rats who receive less than 1 hour of affection per day will demonstrate more aggressive behaviors than those receiving regular affection. So the independent variable is the amount of affection that I'm giving to these rats. The experimental group gets less than an hour, the control group gets the regular level of affection, which in this case is more than an hour. And the dependent variable is the reaction of the rats. In this case, more specifically, the aggressive behaviors being displayed by those rats. Once again, all of these have been outlined in a simple statement, which I've called my hypothesis. Now, I'm also going to ask you to be thinking about experiments. And this is in part because you're going to be designing some experiments when you're in lab during the course of this class. So let's take a look at our first hypothesis, right? The administration of that anti-cancer drug and the volume of the tumor. Some controls could be the type of cancer that I'm testing on. Remember that experimental controls are kept the same between groups. It would make sense to test my drug versus the placebo on two groups that both have the same type of cancer, because a drug might have a different impact based on that type of cancer. I might also control for patient age, the severity of the disease, diet, additional drugs they're on, so on and so forth. These controls aren't necessarily outlined in my hypothesis, But when I'm designing my experiment, I have to think about those experimental controls and make sure that those are kept the same between my experimental group and my control group. When I look at my rats experiment, the type of affection, the type of housing, the type of food, the frequency with which they're fed, those could also be experimental controls. One other one that you might think about, students who eat soup every day will get sick sooner than students who do not eat soup. So controls could include the type of soup, the number of times soup is eaten, the amount of soup, other foods eaten, exercise, age, health. There are an unlimited number of potential variables. But the experimental controls are about identifying the most important of those and keeping them controlled so that they don't influence the outcome. They don't influence what we measure with our dependent variable. Now, let's talk about what you're measuring. There are two types of data. Qualitative data is subjective. You guys probably think of this as things like describing color, so on and so forth. But in truth, color can actually be a qualitative measurement where we're measuring the actual specific pixelation and various different kinds of things. So qualitative versus quantitative is very simple. Qualitative is subjective, meaning two people might provide different qualitative measurements. But quantitative can be measured and indisputable. Length, volume, percentage, absorbance, so on and so forth. Both of these are reasonable and both of them may be included when you're doing materials in lab. But in general, we tend to report that data differently. Pay attention as you complete your writing assignment over the course of this semester, because you would probably be more likely to present qualitative data differently than quantitative data. And that starts by identifying the type of data you're collecting. Now, once you've collected that data, you need to determine whether your hypothesis is supported or unsupported. I want to remind you that a hypothesis can never be proven true because you cannot account for every single variable. And that would be the only way to prove it was true, is that there's not some other variable which is causing the change that you're observing. So we're going to use two other terms, supported or unsupported. If a hypothesis is supported, experiments are consistent with that hypothesis. And if it's unsupported, they're inconsistent. If it's unsupported or supported, we need to rework our hypothesis, report those results, and then repeat by developing a new hypothesis that will further our understanding. Now, let's take a look at how we apply the scientific method. Here's an example which comes straight out of your textbook. Let's do an experiment to try and explain why these mice have different colors. They're both the same genus and species. However, these two mice look different from one another. So here's my example of a hypothesis. Are white mice less likely to be eaten by dinosaurs than brown mice? So you tell me, do you like my hypothesis? I want you to stop and think about this hypothesis for a moment. Now, here's what I would like you to do. Log into Socrative. Remember that our room number is BIOL 1202S. And I'd like you to write your own hypothesis. And I'm going to ask you 7 questions about this experiment. So pause the video here and work your way through this experiment, answering Socrative questions as you go. Now, Socrative is not about getting answers right. As a matter of fact, I'm not even going to look and see which answers you got right, because I'm not testing you right now. What I am doing is providing you an opportunity to test yourself. So here's what I want you to do. Put your notes away. Close your textbook. Try this out. See what it's going to be like when you get an assignment or an exam that asks you questions like this. If you get it wrong, it means you should be going back and reviewing your textbook. Maybe even rewatching this video. Socrative is not about getting something right. It's about seeing whether or not you understand. So put it all away and try the next 7 questions while you pause this video. When you're done, Go ahead and restart this video and I'll go through the answers with you so that you know what you're looking for. All right, now remember, you're not being graded on accuracy. So don't worry if you got any of these wrong. It's not a big deal. But do make sure that you go back in your notes and star those sections because it means you need to review them before the exam. So when you wrote your hypothesis, did you write something like this? The color patterns in the mice evolved as adaptations that camouflage the mice to protect them from predation by visual hunters. My hypothesis is testable. I have visual hunters that I can use. It's falsifiable. I actually have these things, I can do them. I can prove or I can unsupport, right, this hypothesis. I can go through and see that the data is not supportive of what I've written here. I've also identified the independent variable, which in this case is color patterns. And that my dependent variable, the thing I'm measuring when I change those color patterns, is something I can in fact measure, the predation rate by visual hunters. So here's the experiment that I have designed. The group built 2 habitats. By the way, this is a real experiment that was done by scientists. They built a white habitat, which is a beach habitat, and a darker inland. They also took 2 mice, one of each color, one white and one brown, and they released them into the habitat along with a hungry owl, which, by the way, is a visual predator. The first mouse that was eaten in that habitat was recorded. So which of these habitat color combinations would serve as control groups? Well, controls are all about things that are not being changed, right? This is like the closest to nature as possible. So A and D would actually both be control groups in this particular example because the mouse color actually matches the habitat color. The independent variable, I will remind you, is something that's being changed by the researcher. In a good experiment, only one thing, one factor will be changed by the researcher. If you change more than one factor, you can't identify which one is responsible for changing the dependent variable. So in this case, the color of the enclosure is going to be our independent variable. And remember, there's only ever going to be one independent variable. The dependent variable is what we measure, which in this case is the color of the mouse, which is eaten first. So our hypothesis generated the following experiment, and here's the data. We have in the beach habitat light and dark mice, and in the inland habitat, that's the light colored habitat, light and dark mice. So true or false, our hypothesis was proven correct. This answer will always be false. The reason being because you can never account for every single variable. So we would only ever say that this hypothesis is supported or unsupported. True or false, the data is quantitative. In this case, I would argue no. This is true, right? This is quantitative data because you are measuring the number of mice that you're capturing. So even though there's some aspect to this owl who's making a decision here, because we are actually counting numbers and determining the percentage of these individual mice, this is going to be quantitative, quantities, numbers, measurements, indisputable values that we can use. So what would be the next step in your scientific inquiry? Well, quite simply, once you've reported these results, you would repeat this process by developing a new testable hypothesis that you could use. All right, thank you guys, and I will see you for our next lecture, which will be on Chapter 2 and Chemistry and Water.